I burn myself to provide fuel to life, generated through gluconeogenesis by my friend, engaged in the synthesis of lipids, amino acids, and I deranged in my duties due to the diabetes mellitus. Guess who I am? I am the official spokesperson of carbohydrate metabolism, glucose. Carbohydrates are the major source of energy for the living cells, as carbohydrates are the first cellular constituents synthesized by the green plants during photosynthesis from carbon dioxide and water on absorption of light. Thus, light is the ultimate source of energy for all biological processes. Major pathway of carbohydrate metabolism are glycolysis, citric acid cycle, that is also known as Krebs cycle, gluconeogenesis, glycogenesis, glycogenolysis, hexose monophosphate shunt, uronic acid pathway, galactose metabolism, fructose metabolism, and amino sugar and mucopolysaccharide metabolism. First, we'll start with glycolysis. Glycolysis is derived from Greek words glykos means sweet or sugar and lysis means dissolution. It is a universal pathway in the living cell, and the complete pathway of glycolysis was elucidated in 1940. This pathway is often referred as Embden-Meyerhof pathway, that is EM pathway, in the honor of two biochemists who made a major contribution to the knowledge of glycolysis. Glycolysis is defined as the sequence of reactions converting glucose to pyruvate or lactate with the production of ATP. And all reaction of glycolysis takes place inside the cytoplasm. Next, we shall study about importance of glycolysis pathway. It is the only pathway that is taking place in all the cells of the body. Glycolysis is the only source of energy in every erythrocytes. In exercise, when muscles tissues lack enough oxygen, anaerobic glycolysis forms the major source of energy for muscles. The glycolytic pathway may be considered as preliminary step before complete oxidation. The glycolytic pathway provides carbon skeleton for synthesis of non-essential amino acids as well as glycerol part of fat, and most of the reactions are reversible. In reaction of glycolysis, the pathway is divided into three distinct phases. First is energy investment phase or priming stage. Second is splitting phase. And third is energy generation phase. So first is energy investment phase, in which glucose is phosphorylated to glucose 6-phosphate by hexokinase or glucokinase. Both are isoenzymes. This is an irreversible reaction dependent on ATP and Mg2+. Then glucose 6-phosphate undergo isomerization to give fructose 6-phosphate in presence of enzyme phosphohexose isomerase and Mg2+. In step 3rd, fructose 6-phosphate is phosphorylated to fructose 1,6-biphosphate by phosphofructokinase enzyme. This is an irreversible and a regulatory step in glycolysis. Next is splitting phase. In this case, the 6-carbon fructose 1,6-biphosphate is split into two 3-carbon compound glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldose. Then in next step, the enzyme phosphotriose isomerase catalyzes the reversible interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Next is energy generation phase, in which glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate. This step is an important as it is involved in the formation of NADH plus H plus and high energy compound, that is 1,3-biphosphoglycerate. In case of aerobic condition, NADH passes through the electron transport chain and 6 ATP are synthesized by oxidative phosphorylation. Then, in next step, the enzyme phosphoglycerate kinase acts on 1,3-biphosphoglycerate, resulting in the synthesis of ATP and formation of 3-phosphoglycerate. This step is a good example of substrate level phosphorylation, since the ATP is synthesized from the substrate without involvement of electron transport chain. Then in next step, 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. This is an isomerization reaction. Then the high energy compound phosphophenol pyruvate is generated from 2-phosphoglycerate by the enzyme enolase. Then the enzyme pyruvate kinase catalyzes the transfer of high energy phosphate from phosphophenol pyruvate to ADP, leading to the formation of ATP molecule. This step is also a substrate level phosphorylation. The fate of pyruvate produced in glycolysis depends on the presence or absence of oxygen in the cell. Under anaerobic condition, that is in lack of oxygen, pyruvate is reduced by NADH to lactate in presence of enzyme lactate dehydrogenase. And the formation of lactate allows the regeneration of NAD+, which can be reused by glyceraldehyde 3-phosphate dehydrogenase, so that the glycolysis proceeds even in the absence of oxygen to supply ATP. Say that energy yield from glycolysis during anaerobic respiration are as follows. Here, 1 molecule of glucose is converted to 2 molecules of lactate. There is a net yield of 2 molecules of ATP. Then 4 molecules of ATP are synthesized by 2 substrate level phosphorylation. Net yield is of 2 ATP. And energy yield during aerobic condition is as follows. Here, 2 NADH molecules generated in glyceraldehyde 3-phosphodehydrogenase reaction, and NADH provides 3 ATP. This reaction generates 6 ATP molecules. Therefore, total ATP generated in aerobic condition is 8 molecules. As we know that glycolytic pathway is one of the body's important metabolic pathways. It involves a sequence of enzymatic reactions that break down glucose into pyruvate, creating the energy source, that is ATP molecules and NADH molecules. Along with that, various inherited defects in enzymes of the pathway may also occur. The most common defect is pyruvate kinase deficiency. Other defect that causes hemolytic anemia include deficiency of erythrocyte hexokinase, glucose phosphate isomerase, and phosphofructokinase. Next, we'll study about regulation of glycolysis. The enzymes, namely hexokinase, glucokinase, phosphofructokinase, and pyruvate kinase, catalyzing the irreversible reaction, regulate glycolysis. Hexokinase is inhibited by glucose 6-phosphate. This enzyme prevents the accumulation of glucose 6-phosphate due to product inhibition. Next is glucokinase, which specifically phosphorylates glucose, is an inducible enzyme. The substrate glucose, probably through the involvement of insulin, induces glucokinase. Phosphofructokinase is the most important regulatory enzyme in glycolysis. It is an allosteric enzyme regulated by allosteric effectors.